Well, let's move on to special senses. Today we'll get through our chemosenses. They're pretty straightforward and easy to understand. We'll talk about the anatomy of the eye and the ear, and we'll start in with vision and hearing tomorrow. Are there any questions over skin or anything so far? All right. So, special senses. Special senses use specialized sense organs instead of just general sensory receptors. So, our special sense receptors are found in what we call specialized sense organs, and the receptors are all located somewhere in our head. So, our chemical senses are the first two senses we're going to go over. So, smell and olfaction. Smell is olfaction, taste is gustation, and both of those are chemical senses. So what type of receptor must they use? A chemoreceptor. So our chemical senses use chemoreceptors. Chemoreceptors only work when our chemicals are suspended in fluid. So for a chemoreceptor to be activated, chemicals must be suspended in fluid. So that's the first thing. Smell receptors are excited by odorant molecules. So smell receptors are excited by odorants. And taste receptors respond to tastants. So what must my odorant be suspended in for me to smell it? In what kind? Mucus. So tastants, what kind of fluid must we be suspended in for a taste bud? Saliva. So which also contains mucus, but it contains other stuff. So that's how we're going to activate our chemoreceptors, by taking those odorants, suspending them in mucus, taking those tastants, suspending them in saliva, and then they will interact with the receptors for each of those in just a moment. So, our organ of smell. So all of our special senses use special sense organs. And the organ of smell is called the olfactory epithelium. I'll wait, because it looks like people might still be writing. Everybody good? Shout no if no. Okay, so the organ of smell is called the olfactory epithelium. And it's found at your superior nasal concha, which is why we looked at all of that stuff at the beginning of class. So the organ of smell is called olfactory epithelium. And it's located in your nasal cavity. It covers the superior nasal concha. And it contains what are called olfactory receptor cells. Olfactory receptor cells. So these olfactory receptor cells are bipolar neurons that are covered with what we call olfactory cilia. So we'll say they are bipolar neurons with olfactory cilia. classification is this bipolar neuron. Is this describing its function or its structure? This is the structural classification. What type of neuron is it by function? By function, if we're thinking about, this is our receptor cell that is a bipolar neuron, we actually consider, it would be considered, it's a sensory neuron. And so this is one of the only two places that we find bipolar neurons in the body. They're pretty rare. There are supporting cells that surround our olfactory receptor cells. And at the base of the epithelium, we have olfactory stem cells. So not very many neurons regenerate. Some places they do, and this is one area that they do. Your little bipolar neurons here that are acting as our olfactory receptor cells do undergo mitosis and get regenerated. So what this is showing you is that as you inhale air, it's going to go up your nasal cavity, and these nasal concha create that turbulence that is going to whack the air molecules into this mucus, and odorants then get suspended in it. So what you can see now from this picture is that these olfactory epithelial cells are reaching up, or this epithelium is reaching up through that cribriform plate of the ethmoid bone. So those are called olfactory foramina, and that we see in the cribriform plate, and they're going to synapse up here in the olfactory bulb. So our olfactory receptor cells are these cells right here. They've got supporting cells. Here are their olfactory cilia sticking out in the mucus. So odorants get suspended in that, and they can respond. They're going to transmit this message next to these cells called mitral cells. And the mitral cells are going to send their axons throughout the rest of our odorant pathway. So we'll say that our bipolar neurons are going to synapse on mitral cells. And these mitral cells are also bipolar neurons, and they're extending their axons away in the olfactory tract. So our mitral cells, our axons continue to form the olfactory tract. All right, any questions about olfactory epithelium? I'm just going to start at the top. What? Questions? It's to form the olfactory tract. Any other questions? Is everybody good on this? Okay. So, in order to smell a substance, it has to be volatile. So it has to be in the gaseous state, and it's got to be suspended in that mucus again. So that's all we need to be aware of, is that it's volatile, which means that it's in the gaseous state, and it gets suspended in mucus. So the activation of our olfactory sensory neurons is pretty complex. So our dissolved odorants are going to bind to these receptor proteins that are going to open these things called, cation channels, and will generate receptor potentials that are going to go through this whole thing, activation of these G proteins. So it's pretty complex, and I'm not worried that you memorize every single step. We just need to be aware of a few things. So we've got to be in gaseous form, and we've got to be suspended in mucus. We've already talked about that. Our dissolved odorants are going to bind to receptor proteins in our olfactory cilia membranes, which open, so binding opens a cation channel. So is that going to be depolarizing or hyperpolarizing? Depolarizing, because the cation channel is going to let sodium in and potassium out, but sodium in to a greater degree. So what it does is generate what we call a receptor potential. And this receptor potential is going to allow for an action potential to be conducted to our first relay station in the olfactory bulb. So we're going from our first order neuron is that bipolar cell, or our olfactory receptor cell. So our olfactory receptor cell is our first order neuron. And our mitral cell is our second order neuron. Our mitral cells are going to send their axons directly to the frontal lobe. So just kind of be aware of that we aren't, to have our conscious perception of smell, we don't have to have a 3 neuron chain. We are going to have a three neuron chain in other parts because we're going to send, our mitral cells are going to synapse in other areas. We can go through our thalamus to get to our hypothalamus and these other parts. So just kind of be aware of that. But for our first, our ability to recognize and be aware of it, we only have to go through 2 neurons. And so our mitral cells are our second order neurons, and we'll say that they synapse in the primary olfactory cortex. Where is that? The primary olfactory cortex. It's actually in our temporal lobes. Your frontal lobes are involved to some degree. Smell goes through crazy kind of pathways and does a bunch of stuff. That's why it's so potent for being able to elicit memories and recall. So we'll talk more about that in a minute. So we'll smell transduction. I guess that's another thing that I should point out, is transduction is converting one type of message to another type of message. So what we're doing here is we're taking a chemical message in smell and converting it to an action potential message, so an electrical message that's going to be conducted throughout the rest of the pathway. So in order to do this, the odorant is going to bind to a type of receptor. These are called G protein coupled receptors. They get that name because the receptor is going to activate a protein. In this case, the G olfactory protein. G proteins get activated when the odorant binds. And then they go off and they activate some other receptor or some other protein. So our receptor activates G olfactory. G olfactory is now activated and it's going to go activate this other protein, adenylate cyclase. Adenylate cyclase starts kicking out this second messenger called cyclic AMP. It's a second messenger because this is the first messenger. And now I'm going to be producing a bunch of second messengers that are going to cause a cascade of changes inside the cell. In this case, what I'm doing is opening up this sodium and calcium channel that's going to allow for super depolarizing events through that channel, because now I'm bringing in 3 positive charges. You know, you could kind of think about it that way. So what do I want you to know from that? That we're transducing an odorant message to a chemical message. That's what you need to know. You don't need to worry about each of these steps. The reason I showed you this is because second messenger systems are going to come up over and over and over again. And this is how G proteins work. So just kind of be aware of that. So our olfactory nerves, or those olfactory receptor cells, these bipolar cells, are going to synapse on the mitral cells that are hanging out in the olfactory bulb. Did anybody note a difference in the size of the olfactory bulb on the actual sheep brain and the models? You should have noticed a difference, which is that on the sheep brain, their olfactory bulbs are huge. They're like that big. And on all of our models, our olfactory bulbs are barely bigger than the olfactory tract. So what does that tell you about how important the sense of smell is to a sheep versus a human? Incredibly. Animals rely on their sense of smell a lot more than we do. So they've got really big olfactory bulbs. So our mitral cells, we could just say, are the second order neurons that are going to form the olfactory tract. So the synapse is occurring in these structures we call glomeruli. I'm not going to test you on the name of glomeruli or anything like that. Axons from our neurons with the same receptor type converge. The mitral cells then are going to amplify, refine, and relay the signals. Impulses are going to lots of places. We're going down from our olfactory tracts to the olfactory cortex. Some of the information is getting sent to the frontal lobe. Some passes through the thalamus first. So this is kind of a weird thing. To get to your primary olfactory cortex, our olfactory cortex is where we perceive smell. By coming here, interpreting and identifying, by coming to our frontal lobes, this is where we're going to give more kind of characteristics to smell, and kind of where we're bringing it through and making more kind of complex things about it. As here, so some information is sent to the hypothalamus. This is going to help with activating things like salivation. The amygdaloid bodies are part of this region that is involved in long-term potentiation of memories, and they are really potent for fear. So that's just kind of something interesting to be aware of. Other parts of the limbic system. Limbic system controls emotions, right? Limbic system is involved in the formation of memories. So we've got the hippocampus is there and all of that. So by being processed in these structures, this is what gives smell such an emotional impact. And because all of those structures are deep where memory is formed, smell is the most potent trigger of memory. So I could taste something, and that's also pretty good for memory, but if I just like taste a gingerbread cookie, it's different than if I walk into a house that is full of the smell of gingerbread cookies. That's likely to bring me back to my grandma's house where I made gingerbread cookies. Where if I just eat one, it's not necessarily eliciting the same thing. It's definitely touching one isn't going to do anything. So because of this complex processing, smell kind of has a lot of effect, a bigger effect for you, maybe in your everyday goings on than our other smells, our other senses. So that's going to be beneficial for a couple things. Like what? What is it beneficial to have smell be so potent for? Why is the sense of smell important to your survival?
It can warn you.
It can warn you. So things that smell noxious, you avoid. So if something's poisonous or going to be toxic, you'll avoid it. If something smells delicious, you're likely to go to that again and eat more of it. So that's kind of how it's helping with that. Any questions? That's the level of detail you need to know for the sense of smell. The chemo sense